Hi everybody. For this weekend you're going to be continuing on with the notes that we took in class, and I want to start by going over the two small labs that we did in class. And so if I already went over this with your particular class, you can either listen to this again or you can fast forward to where the new stuff is, okay? It's up to you guys. Alright, so remember we had iodine in this class, and we had starch here, and then we had our semi-permeable membrane, which is this, which was supposed to represent a cell. And we were trying to figure out what is able to diffuse. All right, so remember, diffusion is just the random movement of molecules. And so this starch is kind of moving around in the water, and the iodine's moving too. We just can't see it. So what happened was our bag turned purple. So what that means was inside the bag, iodine mixed with starch, which means iodine can fit through the bag. Now, why is it that iodine can fit through, but starch did not get out? And that has to do with size. Iodine's a much smaller molecule. If you remember starch, that's a carbohydrate. So it's going to be made of lots of these monomers of sugars or monosaccharides. And I'm not going to draw all of them because there's hundreds. And so if you think about it, that's a huge molecule to fit through such a small little pore in this membrane. Iodine, on the other hand, that's just two elements. It's I2. And so that's going to be able to go right through. And so some of the iodine's going to wander in here and mix with that starch. And then it's not going to come back out again once it's actually linked to the starch. Okay? All right, so this is just an example of a cell's semi-permeability, okay? And diffusion. In our next experiment, we looked at these Allodia plant cells. And so in your diagram, make sure you label a couple things. One, this is the cell wall. Okay, and cell walls are like a box around plant cells, okay? So imagine this box here, okay? And within that box, there's actually a cell membrane inside of it, okay? So it's like the cell, which is squishy, is shoved inside this really rigid box, okay? And the cell is so full of water here. This is the fresh water, okay? The cell is so bursting with water that you can't even tell the difference between these two, the cell wall and the cell membrane. All right, and so you probably saw some scattered little dots, these green things. Those are what we call chloroplasts. Okay, so make sure you label one of those on your diagram. And inside the chloroplast, there is chlorophyll. Oops. And chlorophyll is what actually does photosynthesis.
In the dining hall as light refreshments are being served right now, courtesy of our Parents Association.
Okay. Excuse that announcement. Okay, and this is the salt water. All right, so the salt water, you probably saw all these little green chloroplasts sort of clump together. At least you should have, hopefully. And when the reason why they're clumping together is, look, the cell membrane is actually right there, okay? So you can actually see the cell membrane now. Oops, it's not letting me write over there. Membrane. Okay, so there we go. So how did the cell get from this big to this big? All right, so you got to imagine there's salt all around. And polar things do not get through the cell membrane very easily, okay? Remember, polar big things don't really go through there. So what's going to happen is, though, water is going to escape, all right? And if you've ever eaten something really salty, you know that you get thirsty. And that's because all your water is going to go and stick to the salt. Because salt is polar and water's polar, so they like each other. And so the water inside the cell goes and is attracted to the salt. And once it's stuck to a salt, it can't go back in. And so the water ends up hydrogen bonding to the salt and not re-entering the cell again. And so the cell ends up shrinking like this. And that process is called osmosis. Okay, and osmosis is just when water moves across a membrane. Okay, and I also want you guys to remember that water follows solutes. Okay, so salt is a solute. A solute is anything that dissolves in water. Okay, so anywhere where there's solutes, water is going to get attracted to it and move that way. Okay, and so let me give you another example of this. And you want to open up to your composition notebooks, which you have this diagram right here. Okay, now let's look at this situation. This is what we did to our Lodia cell, but this time that's a red blood cell. Okay, so these are pictures of red blood cells. All right, and they're put in different solutions. This one is really, really salty, okay? And anytime something has a lot of solutes, more solutes than anywhere else, we say that's hypertonic, okay? So go ahead and write down this means more solutes. Okay, so you got to imagine there's all this. You can think of it as salt or anything that dissolves in water. It could be sugar, okay? So that's all going to be there. And see this big arrow out? That means water is leaving. And the arrow in means that not very much water is going in here. Okay, so when the water leaves and is attracted to the solutes, the cell shrinks here. Now on this side, in the middle, this is a hypotonic solution, which means there's less solutes here. And this is just fresh water here. Okay, so that means hypotonic, there's less solute. There's no solutes here, so that must mean there's a few inside. And so water is going to get attracted into the cell, so water enters. And some of the water is going to leave too, but not as much as it's going to come in. So overall, the cell is going to blow up, and it actually would blow up so much that it would burst. So we can't ever put our cells in just fresh water. It helps to have a little bit of solute in our liquids. Okay, now isotonic, this means the same number of solutes. And in this case, the red blood cell looks normal. The same amount of water goes in, same amount of water goes out, because there's the same concentration of solutes. Okay, so that's why our blood plasma has some solutes in it, and so does the inside of the cell, and it's equal, so our cells stay the same size. Okay? Now, let's look at what the plant cell looks like. It's a little different here, okay? So let's look at what we have here. This is hypertonic, so this is in really salty salt water. You see the water leaves and the cell shrinks here. All right, here it's isotonic. Not too great. It's still a little, we say flaccid. That means in this case the plant would be kind of like droopy, okay? Turgid, that's when water goes in and the plant stays upright. You don't have this diagram, so don't worry about writing it down. I'm just showing you what it looks like with a cell membrane around it. So this is like your Elodia experiment. Okay, but now let's move on to something called active transport. So active transport is when we actually need energy, okay? And the energy in the cell is called ATP, all right? So you just kind of need to know that there, all right? Now, so here's our cell membrane, and here's a protein that goes all the way through, all right? So notice we're taking these squares and stockpiling them over here, okay? Normally what would end up happening if we didn't have any ATP is we'd want to go from where there's high concentration to low. But notice we're going opposite, okay? We're trying to get all of them on one side. And anytime you try to stockpile something on one side of a membrane, you're going to need some energy to do that. Okay, so this is needed for moving substances from low to high concentration. Okay, and so we're going to need energies to move that against the flow or against the concentration gradient, we say. Okay, so this is what we call a protein pump. And there it is. All right, another example of active transport. Oops. Uh-oh. Let's see if I have this diagram. Uh-oh, looks like I don't have the diagram. Let me add in a slide here. Okay, so you have a diagram of, oops. Here we go. Okay. In your notebook, you have a diagram of what's called endocytosis. And there's also something called exocytosis. Okay, and this is for very large things, okay? Anything that is too big to go through a protein. All right, so let's say this is our cell, and this is something outside of the cell that needs to come in. Okay, so let's say this is like, let's say it's an amoeba, a single-celled organism, and it wants to eat this bacteria. Okay, so let's say this thing is going to eat this thing. What it's going to do is actually, I'm going to draw it again, it's going to wrap the cell membrane around it. Okay, so it's going to do this sort of thing. And it's going to go all the way around until it brings that thing inside. Okay, so this is called endocytosis because we're going to bring this thing in. If you imagine the arrows going the opposite direction, that is going to go move out and it'll be released and the cell will go back to normal. Okay, so this way is endocytosis. If we go this way, it's exocytosis. Exocytosis. Okay, so you can also imagine this happening with like a lot of little molecules. Okay, let's say if this cell wanted to bring all that stuff in, it could do that. Okay, so it can take in one big thing or lots of little things. Anytime we need to bring in a large volume of stuff, that's what we would do. Okay, so those are all the different kinds of transport. Let's just review a little bit. So let's go back here. All right, so our cell membrane is semi-permeable. Things can either simply diffuse through, that doesn't require energy, that's passive. It can also go through carrier proteins, all right, and that's still passive. It's from high concentration to low. And anytime water moves through a cell, that's called osmosis. That's also passive, that doesn't require energy. And then, then there's active transport. This needs energy, so this is when we go the opposite direction. We stockpile materials. We need ATP for that. And also when we're doing endo and exocytosis. All right, hope that makes sense, guys, and I'll see you guys in class.